Carbohydrates metabolism. This refers to digestion and absorption of carbohydrates. The process that is collectively and primarily involved in the digestion and absorption is known as metabolism. Now, the clip in the middle describes the sum total of all the chemical reactions that is involved in our body, and that it's with respect to the food intake or the food that we eat as indicated in the body. This biochemical reaction is important in maintaining the living state of the cells and organism, that is to sustain the life. Now, there are two types of chemical breakdown, and this can be categorized into anabolism at the left side of the video. You may notice that there are smaller units of molecules, or the monomers. And this will require energy to grow and build into a complex molecule or to a polymers. And at the right side of the video, we have the catabolism, and this is the breaking down of a complex monomer or a complex molecules as it uses energy to break down into a simple molecules or to a monomer. Carbohydrates digestion. This digestion involves the conversion of polysaccharides and the disaccharides into a monosaccharides. Let us initially evaluate the pathway of food particle into the body. As the food enters into the mouth, this is where mastication or chewing or grinding will take place. And there will be aid of salivary enzyme from the salivary glands in order for partial degradation of food to occur. And then once the food has been churned into food particles, it will descend down into the esophagus, that long part descending down to the stomach. And in the stomach, this is where the neutralization of salivary enzyme will happen. And this serves as a mixing bowl for the food. And further, it will propel the food into small intestine, where absorption will finally take place. And this is with the aid of pancreas and liver secretion. Now, basically, the carbohydrate source refers to the food. And in this case, hamburger is our food source. It is from this food source where polysaccharides are coming from, and that is in the form of starch and the glycogen for the polysaccharides. And for the disaccharides, it will be in the form of sucrose, lactose, and the maltose. As mastication or chewing grinds the food particle into the mouth, there will be an enzyme from the salivary glands, and we call that enzyme as the amylase. And this amylase will degrade the polysaccharide that we mentioned, such as the starch and the glycogen. And there must be an activation of chloride ion at an optimum pH of 6.7. Now, other than the polysaccharides, the disaccharides will be degraded by the dextrine, which is collectively we call it as the disaccharidase. And this will be in three forms. If the disaccharide sucrose is to be degraded, an enzyme is a sucrase. If the disaccharides maltose is to be degraded, then the enzyme is maltase. And if the disaccharides lactose is to be degraded, the enzyme will be lactase. All of these, both the polysaccharides and the disaccharides, must be converted into a monomers, or what we call the monosaccharides. Oxidation. Oxidation has a role in carbohydrates digestion. And in order to analyze that, we have to look first into the importance of glucose. It is said that glucose is on top of a cellular menu. That means this is the prepared consumable of a cell, because this is also known as a blood sugar and the major breakdown product of carbohydrates digestion. Oxidation plays a role particularly in the making of ATP in most body cell, and that oxidation is the loss of electron during a reaction by a molecule, an atom, or ion, as depicted in the middle clip. From a ring chain of a molecule of a monomer, there will be an interaction where there will be loss of electrons into another molecule or atom. Now, glucose is the most important food, and as well as the most abundant food available in the CSF, or cerebrospinal fluid. And that brain consumes the most of glucose, as indicated in the clip at the right side. Brain continuously depletes the glucose of the body. Now, when a glucose is oxidized, meaning when the glucose is combined with oxygen in a complex chemical reaction, further the glucose is broken down into two, particularly the carbon dioxide and the water. And it is this chain reaction that occurs in the presence of oxygen. And with this, it is responsible for the deterioration in the quality of food products. Now, in our later discussion with anhydrase and carbonic anhydrase, the said byproduct of oxidation into carbon dioxide and water will play an important role. The carbohydrates absorption. As we look at the left anatomical illustration, there are organs primarily responsible for the carbohydrates absorption, and that is the small intestine, which is considered as the major site for carbohydrates breakdown. However, the chyme must first be mixed at the stomach, and that in the stomach it has a pH of 3, which is highly acidic, and this acidic nature will stop the salivary amylase from working. The presence of hydrochloric acid will also cause the hydrolysis of monosaccharides, from which we have learned from our previous discussion that hydrolysis is the process of breaking down the polysaccharides or the disaccharides into a smaller component of monomer. And gastric juices is present in the stomach as well, but it carries no enzyme to break the glycosidic bond. So therefore, it must be propelled down to the small intestine. Now, If you may notice with the clip in the middle, it depicts the entirety of the small intestine, which all starts from the duodenum, the first part. Now from there, the pancreatic juice is being mixed up and released, carrying pancreatic amylase, which will further require chloride ion to function and has a pH of 7.1, and from a highly acidic hydrochloric acid of stomach, it is now turned into a neutral to semi-alkalinic. This will convert the starch and glycogen to disaccharides and oligosaccharides. Now let's take a closer look and a microscopic look at the right side of the slides, from which the right side depicts a microscopic appearance of microvilli inside the duodenum or small intestine, from which you may see there are degradation of the polysaccharides in the ring shape molecules and even the straight line molecules. Now, as all of these polysaccharides, disaccharides are being broken down, it will be aided by the brass border enzymes. Now, these brass border enzymes are also called as the disaccharinidase, from which this includes the dextrinase, the glucoamylase, the lactase, the maltase, and the sucrase. These are all enzymes that will speed up into a million folds the process of digestion and absorption. Now, once the food is being converted into a monosaccharides, this will be absorbed further by different processes. And the processes are the passive diffusion, the active transport, and the facilitated diffusion. From which also, from our previous discussion, we have learned that passive diffusion is the movement of molecules from a greater area of concentration to a lesser area with no energy required. And exactly this is the opposite of active transport. And facilitated diffusion will require a carrier protein to absorb all of the monosaccharides or the monomers that is being absorbed by the microvilli of the small intestine. Now, as you may notice, the digestion all started from a polysaccharides, and then it is broken down further into a disaccharides and oligosaccharides. And then at this point of small intestine absorption, it is now at the level of a monosaccharides. Now, in the next slide or in the next discussion, we will look into what are the roles of accessory organs of digestion and what are their contribution in the further degradation and absorption of the carbohydrates. Accessory organs of carbohydrates absorption and the digestion. Two main organs play a role in absorption and digestion of carbohydrates. And these are the liver and the pancreas. Now, liver is said to be the major site for converting excess carbohydrates. So when you have eaten too much, there will be an excess of carbohydrates. Liver will take care of that. And of course, if there is an excess, the liver will store it. And this will be in the form of glycogen. And whenever there is a need, or when a need arises for this glycogen to be released, then the liver will take care of it. And of course, it ensures constancy of the glucose level in that case. Now, having said that, let's take a look at a glycogen. A glycogen is said to be a stored complex chain of glucose. This is a converted glucose and stored in the liver. And to achieve that, to store the glycogen, it must undergo a process called glycogenesis, where the glycogen is being formed from excess glucose. And the opposite of that would be the glycogenolysis, where if the glucose is already stored in the liver and the time needs for it to be extracted or be converted into a glucose again, then the biochemical breakdown of glycogen to glucose is called glycogenolysis. Now let's talk about pancreas. Pancreas is nestled, if you will look at the lower picture at the left, pancreas is nestled posteriorly and inferiorly to the stomach. And you may notice that the head of the pancreas, that bigger part of the pancreas, is having an intricate harmony with the duodenum, which is the first part of the small intestine. And there forms a connection from the pancreatobiliary duct to the ampulla of Vater, where they tend to enter into the small intestine through the duodenum. Now, the pancreas is classified as both exocrine and endocrine gland. Exocrine in the sense that there is a need for a duct to release the enzyme. And the enzyme to be released are the amylase and the bicarbonate. However, the endocrine part of the pancreas is by the release of hormones via blood. And the very familiar hormones that is being released by pancreas is known as the insulin and the glucagon. Now, insulin is necessary if there is so high blood glucose in your body, and that is in cases of hyperglycemia. And glucagon is the opposite of that. Whenever there is an increased blood glucose, it increases the blood glucose when it gets too low. That is in order to release the glycogen. The pancreas completes the digestion of starch and the pancreatic amylase and the brass border enzymes. And the pancreas creates a pH of 8, which neutralizes the acidic chyme. Now, let's take a keen look on how the pancreas releases the enzymes, amylase and bicarbonate, and how the liver releases the bile together at the ampulla of Vater. The lower site, the lower duct is the release of amylase and the bicarbonate, where the upper side of the duct releases the bile. Together, it is being released at the ampulla of Vater. And through this, the absorption and digestion in the main site, which is the small intestine, is being made possible through the help of accessory organs, particularly the liver and the gallbladder. The enzymes. Let us take a look at the role of enzyme as a catalyst in carbohydrates absorption and digestion. Enzymes are multiplier of the rate of chemical reactions. That means it speeds up the chemical reaction in carbohydrates by at least 1,000,000 folds. That is way too fast. While the substrates are bound, in this case, our substrate is the polymers or the polysaccharides to an enzyme. The polymers or the polysaccharides will have to undergo structural changes, and that after the effects of enzyme, then it will produce a new product. Now, if we will look at the structure of enzyme right below the first paragraph, the enzyme is made up of amino acid, which is the building blocks of proteins, and this is being synthesized by the body. And that the enzymes are more than 1,000 in our body, and only a handful of this enzyme are being used in the carbohydrates absorption and digestion. Now, enzymes are still reusable, and the cell will need only a small amount of each enzyme. And there is a rule in biochemistry with respect to enzyme. No enzyme, then no reaction. Now, without an enzyme, a biochemical reaction would occur either far too slow to sustain a life. Now, let's take a closer look at the clip at the right side. It portrays a biochemical catalytic action of enzyme onto a sucrase substrate, which is a disaccharide. And by hydrolysis, as you may see, there is an addition of water, and it will break the glycosidic bond and will cause the fragmentation. And the fragmentation will be from polysaccharides to a monosaccharides, glucose and fructose. Then the water will be transformed into hydrogen ion. Now, this is a typical or a common example of enzymatic catalytic action in the digestion and absorption of carbohydrates. Anhydrase. Let's take a look into the relationship of anhydrase with the carbohydrates metabolism. From our previous discussion of oxidation earlier, we have learned that glucose is on top of the cellular menu. That means glucose is grossly being consumed by the cell. And as a result, oxidation will cause the production of a waste product after undergoing a complex chemical reaction. Once the glucose has been oxidized, it is bound to produce a waste product in the form of carbon dioxide and water. Now, having said this, the anhydrase is a form of an enzyme that will catalyze then the removal of the waste product. That is a result of carbohydrates metabolism. And in particular, the anhydrase is in the form of carbonic anhydrase. Now, this carbonic anhydrase is necessary in the interconversion of carbon dioxide, which is a waste product of carbohydrates metabolism, into carbonic acid. And together, at the left side of the equation on top, you may see that there is a combination of carbon dioxide and water as a reactant, and it will produce a product in between of carbonic acid. And so much so with the right side of the equation, there is a production of bicarbonate and hydrogen ion, which then translate into carbonic acid in the middle. And what you see on top of the carbonic acid, H2CO3, is an enzyme that will act as a catalytic agent in order to remove this waste product of carbonic acid, which translates into carbon dioxide at the left side of the equation and hydrogen ion at the right side of the equation. This carbonic acid and the carbonic anhydrates can be both found in the following body structures. Number one, in the kidney, it can be found particularly in the renal tubular cell. And this can be found also in the red blood cell. The carbonic anhydrase and the carbonic acid can be found also in the gastric mucosa, particularly in the gastric parietal cell. And finally, This is also present in the respiratory system, the alveolar sac. Now, the bottom line of this is that the waste product from carbohydrates or glucose metabolism must be expelled out, either through the renal system or either through the respiratory system. As what we have learned, from our pre-H discussion, these are the three line of defenses when pH is being altered and deranged in the body, either by the buffer system, or it can be excreted through a urine if that is either hydrogen ion or the bicarbonate, or either it can be expelled through exhalation in the respiratory system through the alveolar air sac, from the blood through the alveolar air sac. And that plays the role of anhydrase, particularly the carbonic anhydrase, in carbohydrates digestion and absorption through waste product removal. Once again, you may pause the video. Let us assess and check your comprehension. That's all, folks. This has been your Nurse Radiologico Guru saying thank you for watching and enjoy studying.